Hi everyone, I'm Margaret from Clermont County Public Library. Thank you for joining me today. Today I'm going to be doing a demonstration of how to extract DNA from strawberries using some common household ingredients, so you may already have these on hand. If you'd like to follow along with me as I do the demonstration, you're welcome to do that, and I'm going to put up a list of ingredients here in just a moment. You're also welcome just to observe and to see how this process is done. Before we actually get started with how to extract DNA, I wanted to just very briefly cover what DNA is, which a lot of you may already know. But DNA is a molecule that's inside of our bodies that has all of the instructions that our bodies need in order to grow and to develop. So we have DNA in all of our cells, and it has those instructions for development. All right, and then another thing that I needed to go over was just a little bit of safety information if you are going to be following along at home. First, please do make sure you have an adult to do this with you, because although they're household ingredients, some of them are, they're ones that you need to have adult supervision for. Also, we're going to be pouring liquids, and that poses a special safety risk. So today I'm going to be using eye protection, and I would recommend that you use some sort of eye protection as well, just to help prevent splashing in your face or in your eyes. And then also, I just wanted to show you the proper technique for pouring liquids, and that is to make sure that you have your cups or whatever you're using held out away from your body as far as you can while seeing what you're doing, and make sure that they're below eye level. So you want to make sure, it's kind of hard to see from the angle here, but I'm holding them out kind of at shoulder level so I can still see what I'm doing, but they're out away from my body and I won't be splashing anything into my eyes. All right, let's get started. All right, so we're going to go ahead and do the DNA extraction at this point. And as you can see, I've got my 3 strawberries here with the tops removed. I have pre-measured my table salt to half a teaspoon of table salt. I have a tablespoon of dish detergent, half a cup of water, and we're going to mix this together, and it's going to be our extraction liquid. So I'm going to go ahead and pour the water in with the dish soap, and then add in our salt. I've got some chopsticks here that I'm going to use for mixing everything up. Just going to take a second for that salt to dissolve. All right, we are ready to go. We just need to pour in about 3 tablespoons of this extraction liquid in with our strawberries. So I'm just going to estimate about 3 tablespoons. That looks about right. Okay, so then the fun part begins. I just get to smush up these strawberries. I'm going to try to make sure that the pieces are as small as possible. And then while I'm doing that, I'm just going to explain a little bit of why we're using strawberries today. So you may know that in your cells, your DNA is stored in chromosomes, or you have a certain number of chromosomes. Now, chromosomes are not normally visible except for during active cell division. So if you've seen pictures of what chromosomes look like, where you've got the X, that's actually 2 chromosomes together making a pair, and that only happens during cell division. So normally it doesn't look like that. It's just kind of loose DNA in the cells. It's wrapped around some other molecules that are called histones, and some other proteins that help to make sure that it doesn't get all tangled up and keep it from being a big mess. So, but yeah, it's usually kind of loose and not visible under a microscope, except for during cell division. Now, as humans, we have 23 chromosomes. So we have 2 pairs of those. We have 46 total chromosomes in our cells. Plants are really cool because they, instead of having just 1 copy of every chromosome, sometimes they can have, or 2 copies per cell, they can have many copies of their chromosomes in their cells. So they have lots and lots of DNA. And this is called being polyploid. And in the case of strawberries, they are polyploids. So they have lots and lots of copies of their chromosomes. So they just have a ton of DNA in their cells. And that's why we're using strawberries today, because there's so much of it, it's going to be easier to actually see it once we've done the extraction. So I still have a few pretty big chunks in here. Let me see if I can get those broken up a little bit more. That's looking pretty good. All right, so what we need to do next is actually to filter this to try to get just the liquid, just the DNA in that extraction mixture that we made. So you can use a mesh sieve if you have that at home, or cheesecloth. I actually have paint filtering mesh, which is what I'm going to use in order to filter out my extraction liquid here with the DNA in it. So I'm just going to pour this through. All right, so there we go. Move this out of here, see what we've got. Just going to gently squeeze that. See if you can see it here. It is just dripping in there, kind of making a mess. All right, gonna just continue to gently squeeze this through. And I think that's probably enough right there. All right. So I have made a mess, thoroughly made a mess here, but that is okay. That's part of the fun of this. All right, so you can see I have my strawberry extraction mixture here. It's very pink, especially with that pink dish soap that I had used there. And what we're going to do is we're going to very slowly pour our 70% isopropyl alcohol, which is just something, it's just rubbing alcohol. We're going to very gently pour that down the side of this cup here and see if we can get the DNA to float kind of on the top of this mixture here. So you can see I've had it in an ice bath to make sure that it stays nice and cold. So hopefully this will work out really well. So I'm just going to pour it gently until I've got about ½ an inch of it here on top. All right. And it's kind of hard to see from this angle, but I do have DNA strands that are in the, that are kind of poking up into the alcohol here. I'm going to stop the camera at this point and get a little bit of a better angle for you so you can see what this looks like. All right, here we go. So I've got my mixture here with the DNA extraction. You can actually see this DNA here on top of the liquid. Do you see that moving around in there? There's a ton of it in there, like I mentioned before, because strawberries have so many copies of their chromosomes. They're called octoploid, which means they have 8 copies. You can actually see the DNA here. And there are some proteins that are associated with DNA. I mentioned earlier, histones and things like that. So part of it is that there's the protein on there that you're seeing as well. But this is the DNA here from the strawberry, which is really cool. So now let's talk about what actually happened during that demonstration. What did each of those ingredients do in this?
So first of all, when I was crushing the berries, actually crushing them up with my hands works to break down the cell walls. And then the dish soap in that mixture breaks down the nuclear membrane, and that allows the DNA that's in the nucleoplasm, or the liquid that's inside the nucleus, to be released out into the rest of the solution that we're using. Salt works to neutralize the charge of the DNA molecule. It does have a charge, and that's actually something that helps to make it soluble or able to be dissolved in water. And then another thing that the salt does is to help remove some of the proteins from DNA. And some of those proteins are what helps keep DNA dissolved in water as well. So both of those things, neutralizing the charge and removing some of the proteins, work together to make DNA less soluble in water. And then finally, I added alcohol. And when you add alcohol to the rest of these ingredients in the solution, it makes it so that the DNA just cannot stay in the water. It's really insoluble with the salt and the alcohol together. And then DNA is also not soluble in alcohol, especially really cold alcohol, which I had it stored in the freezer, and then you saw I kept it on an ice bath until I added it in. So it's really not soluble in that cold alcohol. And so the DNA all clumps up and precipitates out of the solution. And that's where we're able to actually see the DNA in the solution.
So I hope that you all really enjoyed that. I know I had a lot of fun. I always love doing this demonstration. It's so cool to be able to actually see the DNA that's inside of the strawberries, and to get that out and kind of be able to experience this yourself. So I do hope that you had fun. I hope you learned something along the way. I really appreciate you joining me today, and I hope we'll see you in the library soon.